Okay, I'm Jeremy Field. I'm from Exeter University, and thanks for inviting me. So I'm going to talk about wasps and phylogenies. So, in the last decade or so, really quite a lot of interesting phylogenies have come out for aculeates. So a phylogeny then, or a phylogenetic tree, shows the evolutionary relationships between different species, or in this case, in this phylogeny, different families. So this paper, for example, by Johnson et al., 2013, I think it was the first one that suggested that the closest living relatives of the apoids, in other words, the sphecoid wasps and the bees, are actually the ants. Before that, I think the ants have been thought of as being part of what used to be called the vespoids. So the vespoids included things like the vespids, pompilids, the scoliids, and the ants. So this Johnson et al. Paper suggested that that old vespoid group isn't really a natural group, isn't a monophyletic group, because there's no common ancestor that includes only the vespoids. In this case, it would also include the apoids. A lot of these phylogenies nowadays are based on DNA data, although they sometimes use a mixture of DNA and morphological data, whereas older phylogenies tend to be based mainly on morphology. Okay, here's another example of an interesting phylogeny that's come out relatively recently. So there are two trees here. The one on the left, this one here, is what for a long time was thought to be the relationships between the different groups within the vespids. So the vespids are up here. So here we've got things like the vespines, so the yellow jackets and the hornets that we get in the UK, the polystines, the paper wasps that you'll have seen if you've gone to Europe or possibly London, stenogastrines, which are the hover wasps that you get in Southeast Asia. And then, so the interesting thing is that the groups that are social have been coloured red, as opposed to the non-social groups like these eumenids here, these potter wasps, which are non-social. So what this older phylogeny suggested was that there was a single origin, perhaps, of sociality in the vespids. So it evolved somewhere here in the common ancestor of all these social groups. Whereas this more recent paper by Piekowski et al., 2018, shows that the stenogastrines are actually the sister group to the rest of the vespids. So that means that that suggests that there are two origins of sociality in the vespids, one down here and one just in the stenogastrines. So in other words, the stenogastrines represent an independent origin of sociality. Another paper you might have seen recently was the one by San et al. 2018. They had a quite impressive data set of something like data from about 200 genes. It was DNA sequences, and I think it was about 170 different species looking just within the apoidea. I think these numbers in brackets are the numbers of species in the different subgroups that they looked at. And one of the interesting things they found, so the bees are called the Anthophila here, and one of the interesting things they found was that the closest living relatives of the bees seems to be a subgroup of the pemphigonines, what we used to call the pemphigonines. So again, that suggests that pemphigonines themselves are not a monophyletic group. They're what's called paraphyletic, so that the common ancestor down here includes the bees and also the phyllanthines, the generalite Phyllanthus and Cerceris. Other groups in their phylogeny, like the crabronids, are not quite the same as what we used to call the crabronids in the more traditional phylogenies. And other papers that came out about the same time, like this one by Peters et al., came up with a fairly similar phylogeny. So anyway, there's been lots of interesting trees have come out, but I don't think I want to go through, keep on just going through them and pointing out what they show. That might be a bit sort of dull. So I thought what I'd do instead is talk now about talk mainly about something you can do with these trees. So I think perhaps that's the most interesting thing about them. And for that, I'm going to focus on one of the groups I've worked on, which are the sphecoids within within the apoids. So the species then are often known as, or sometimes known as thread-waisted wasps, because they tend to have this long, thin petiole that you'll be familiar with. And there are three subfamilies within the Specidae. There's the Scelifreni, which are the mud daubers, include the mud daubers like Scelifron, that you'll have seen again if you've been anywhere apart from the UK. There's the Ammophelines, including Ammophila and Podolonia, that we get in the UK. And then there's the Specines, including genera like Sphex, Prionyx, and Isodontia. They haven't occurred in the UK, although there have been one or two records, I think, of Isodontia and Prionyx recently, so they might be spreading. So anyway, I'm going to focus on these species. So now you're going to see a little video clip now as an Ammophila female coming back to her nest with a prey item. So here's the nest is between these two Yellowstones, and here comes the female. You can see she's individually marked with paint dots on her thorax so that we can recognize her. She's just opened her nest entrance and she's taking out a bit of soil, and then she's going to drag that caterpillar that she brought into the nest. Here she goes, she backs into the burrow, pulls the caterpillar in, and now she's going to lay an egg on the caterpillar. And you might have seen that happen yourself. So in that case then, the female had constructed her nest first before she hunted for any prey. So we call this a nest first strategy. So here's a little diagram of the nest. It's just a short burrow in the soil that gets closed up whenever the female's away. Here's that first caterpillar in the nest with an egg on it. And this is an Ammophila sabulosa female carrying a big caterpillar. And some of these nest first species, they just give their offspring a single large prey item, like some Ammophila sabulosa females do. And here's the Ammophila larva eating that big caterpillar. But other nest first species give each offspring multiple smaller prey. So this is from a nest of a different Ammophila species. And you can see there's about 10 or 12 different small prey items for the offspring. Now, rather surprisingly, though, there are also some prey first species. In other words, where the female catches a prey before constructing a nest. So here's a female, she's got a caterpillar. What she has to do then is she just dumps the caterpillar next to her, or more often she hides it in a bit of vegetation nearby, and then she constructs her burrow and eventually pulls the caterpillar into the burrow. And an example of that is our own Podolonia hirsuta. And there might be a bit an advantage there in that the female doesn't necessarily have to memorize the location of her nest, because she catches her prey and then she digs a burrow. So she might be able to build her nest near to the point of prey capture, and perhaps also reduce her carriage costs. She might not have to carry the prey so far. And these prey first species, though, always provide only one big caterpillar to their offspring. The other thing, so those nest first and prey first strategies I've talked about so far are all what are called mass provisioners. So the point is that all of the food is given to the offspring, is put in the nest before the eggs even hatched. So even in a nest first species that provides multiple prey, she puts the first caterpillar in and lays her egg, and then she gets the rest of the caterpillars very quickly, usually before the egg hatches. So there's no contact between the mother and the larva as it's growing. And that contrasts with the final strategy, which is progressive provisioning, where the offspring is fed more gradually as it grows. And an example of that in the UK is Ammophila pubescens. So what happens in a progressive provisioner then is that the mother constructs her nest, and then she puts in that first prey and lays her egg, as you saw in the video clip. But then she waits 2 or 3 days while her egg hatches and her larva starts feeding on the caterpillar. And then she comes along and does an assessment visit and checks that her offspring has hatched successfully. And only then does she add more prey items, perhaps another 8 prey items. And then only then she closes up the nest permanently and starts on another one. And these progressive provisioners, though, can have more than one active nest at the same time. So in these 2 or 3 days, while she waits for her egg to hatch, the mother might start another nest or 2 elsewhere. So with progressive provisioning, then, the mother interacts with her larva as she's feeding it gradually, and she's got to sort of gauge its needs. She's got to decide whether to give it more food and whether it needs more food. And because she has multiple nests, she's got to schedule her feeds across those different offspring in different nests. And sometimes, amazingly, we found that Ammophila pubescens can have up to 8 different nests on the go at the same time. So she's also going to have to memorize the locations of all the different nests. So progressive provisioning then might be the most kind of cognitively costly or neurally costly of the different. It's quite complex. And one of the things we're actually doing is trying to find out, is that strategy, which we think of as more complex, is it actually more costly neurally? So what we're doing is we're micro CT scanning the brains of different Ammophila species, and we're measuring the size of different regions, like the mushroom bodies, which are thought to be important for learning and memory. And we're trying to see, are those regions larger, for example, in progressive provisioners that have to memorize all these different nest locations than in mass provisioners? And this is some work being done by Phylogenies, who's a PhD. Now, how did these different nesting strategies evolve? And since the 1970s, really, when this book came out called The Wasps by Howard Evans and Mary Jane West Eberhardt, 2 American entomologists, what they suggested in their book is that evolution had gone something like this. So they suggested a stepping stone model for the evolution of these different parental care strategies. And really, the idea was that things went from the simplest strategy, prey first, to the most complex strategy, progressive provisioning. And the logic was, for example, that the prey first strategy is the strategy that's most similar to ancestral parasitoids. So a parasitoid finds a host and lays an egg on it. The prey first strategy is to find a big prey item, lay an egg on it, but constructing a burrow in between. And also there's some sort of logical interdependence between the different stones in the sequence. So, for example, the prey first strategy is unlikely to evolve if feeding multiple food items has already evolved. Because if you're prey first and you caught multiple prey, you'd have to leave those multiple prey lying around in the environment while you went and hunted each prey item. They'd be very vulnerable to scavengers like ants and parasites. And indeed, even prey first species today, even if they catch only one big prey item, tend to get rather heavily parasitized. Also, for example, progressive provisioning seems unlikely to evolve unless multiple prey items have already evolved, because progressive provisioning obviously involves feeding your offspring gradually. You've got to provide multiple lumps of food. Now, on the other hand, these more complex strategies, then they're likely to have these greater, perhaps neural costs going from left to right. So the nest first strategy means you've got to know you've got to remember where your nest is, unlike prey first. And progressive provisioning, as I mentioned, means that you're going to have to gauge the needs of your offspring as it grows, and you can have multiple nests at the same time. So why would you evolve then these more complex strategies if they're more costly? And the answer might be that they also provide protection against natural enemies. Okay, so the nest first strategy then means that when you bring your prey to your nest, you can just put it straight into the nest. You don't have to leave it lying around in the environment like the prey first strategy does. And then progressive provisioning has other advantages against natural enemies. So, for example, lots of specids get attacked, lots of aculeates in general get attacked by miltogramine flies. So Ammophila, for example, tends to get attacked by this genus Metopia. And these miltgramine flies, the females contain in their abdomens, they contain a big batch of eggs that hatch as soon as they're laid. So when a female wasp opens her burrow to take in a prey item, what might happen is one of these flies might nip into the burrow and dump a couple of maggots into the burrow. And if you take the maggots out of a fly's abdomen, like I've done here, and just put them in a drop of saline, they just hatch immediately. And what happens is if a fly drops a couple of maggots into a burrow, they eat the wasp egg, and then they've got the food, the prey items to feed on. So they're kind of brood parasites, cuckoo parasites. And I don't know whether you can see, but here are some little Metopia maggots that I got out, that I sort of dug out of a nest. They've eaten the Haemophila egg, and now they're feeding caterpillars that were meant to be Haemophila larvae. Now, progressive provisioning, though, provides two defenses against these kinds of enemies. So first of all, in this 2 or 3 days, so the mother puts in her first prey item, lays her egg, and then waits 2 or 3 days. During that time, her egg hatches, if it's okay. But if a miltgramine maggot has got in, then by the time the mother comes and does her assessment visit, she'll be able to tell that her egg isn't there anymore, and she'll see the miltgramine maggots because they'll have grown quite a lot. And what she can do then is that she can abandon the offspring if it got parasitized with that first prey item. And that's what progressive provisioning mothers do. Whereas if you're a mass provisioner and your offspring gets parasitized, even if it's the first caterpillar, because you put all the caterpillars in before the egg hatches, there's nothing you can really do, because the miltgramine maggots are still going to be tiny. When you finish provisioning, you won't be able to detect their presence, your egg won't have been destroyed. So you waste a full quota of food on an offspring that's going to fail. The second advantage of progressive provisioning is it turns out that the offspring are immune once the egg is hatched. So in a progressive provisioner, the mother puts in the first caterpillar and lays her egg. And she only puts in the other food items once her eggs hatch. And it turns out that if miltgramine maggots get in with these other caterpillars, because the wasp egg is hatched, the wasp larva is too tough, it's immune to these maggots. Whereas if you're a mass provisioner, you put all the food in before your egg hatches. So 8 prey items means 8 chances of your offspring. Okay, so this stepping stone model then makes some intuitive sense. We go from this sort of simple, neurally cheap strategy forwards in this sequence to the most complex strategy, progressive provisioning, that's perhaps more costly in terms of cognition, but on the other hand, provides the best defense against enemies. So the question is this stepping stone model correct, especially for Haemophilines? So this is part of a phylogenetic tree that we produced for some specids, especially Haemophilines. And it's a fairly well-supported tree, and it generally recovers the 3 subfamilies, okay, as monophyletic groups. So here's the Scelifrini, things like Sceliphron. So up the top here, they are clustered together as a monophyletic group. The Sphecynes, like Sphex, also clustered together, although they come out as paraphyletic here because the Haemophilines are nested within them. And also within so from here downwards, this is the Haemophilines here. And the phylogeny recovers the different genera also as monophyletic groups. So here's Haemophila, Podylonia, Aramnophila, etc. The least well supported is Podylonia, and that sort of agrees also with what sort of morphologists think. So, the tree makes reasonable sense. But what does it say then about this stepping stone model? So here, what these different coloured dots indicate is the nesting strategy that each one of these species uses. So the blue, first of all, are prey first species that get the prey before constructing the burrow. So both of our Podylonia species that we get in the UK, a couple of American Haemophila species, and this Pryonyx here. The red dots are nest first species that provide only a single large prey item. so various Ammophila species and also Podolonia vallida. Also, then we've got nest first species that provide multiple prey in yellow, and that's most of the other non-Ammophiline Sphaecids and also some Ammophila. And then finally, we've got progressive provisioning in green. We've got 3 Ammophila species and this genus Aremocaris. The other thing to notice is that we've sort of reconstructed, if you like, what the nesting strategies were of the ancestors in this tree. So, for example, these 4 Ammophila here that are all nest first multiple prey probably had ancestors that were also nest first multiple prey. And sometimes these reconstructed ancestral states are a little bit uncertain. So here we've got a pie chart shaded partly red for nest first single prey and partly yellow for nest first multiple prey. So there's a bit of uncertainty about the most likely strategy of this ancestor. And generally, we have to take these reconstructed strategies with a little bit of a pinch of salt, because they do depend a bit on the sort of model of evolution that you assume. So anyway, what does this suggest then about the stepping stone model? And the answer is that it doesn't really fully support the stepping stone model. So first of all, the ancestral state for the Sphaecids here isn't the simplest strategy. The prey first strategy is actually this nest first multiple prey strategy in yellow. And that means that there are sort of reversals, if you like, reverse transitions according to the stepping stone model in the phylogeny. So for example, if you look at the prey first species, they have evolved, according to this tree, they've evolved actually from the nest first single prey strategy. So they've sort of gone backwards along the stepping stone model, if you like. And that's true of all of these blue dots for the prey first strategy. There's also some reverse transitions from nest first multiple prey to nest first single prey. So for example, here we go from nest first multiple prey to nest first single prey in Ammophila mediata. And that happens also down at the base of near the base of the tree here. This group of red dots here is reconstructed as having evolved from a nest first multiple prey ancestor. On the other hand, there's some things that do agree with the stepping stone model. So progressive provisioning in green does seem to be a derived strategy. Okay, there's no evidence of sort of going backwards in the sequence once you reach progressive provisioning. And it does seem to evolve from, the nest first strategies. So here from nest first multiple prey. So overall then, the stepping stone model is only partly supported. Okay, and why is that? Well, probably the stepping stone model is a little bit of an anthropomorphism. Perhaps people quite like the idea that things go from simple to complex. It's quite an attractive idea, and from the sort of least cognitively demanding to the most cognitively demanding. But what our phylogeny suggests is that things quite often go in the opposite direction as well. Okay, things don't always go forward in this sequence. Having said that, although the stepping stone model isn't fully supported, the phylogeny does suggest that there is it does agree that there's some logical interdependence still between some of these different strategies. So the prey first strategy, for example, on our tree always seems to have been a transition from nest first with single prey, and progressive provisioning always seems to be a transition from these nest first strategies, usually the nest first with multiple prey. But the point is, the benefits of these more costly strategies like progressive provisioning are going to depend on ecology. So they're going to depend on things like prey availability, which might vary in space and might change over evolutionary time. So if you're in an environment with perhaps lots of small prey, and nest first multiple prey has evolved, but then the environment changes and there start to be lots of large prey, perhaps you'll get a reversal to nest first single prey. Also, the ancestral strategy being this nest first multiple prey is probably the most flexible strategy, and it's the most, it's the commonest strategy in the tree. So it's flexible in the sense that however large prey are, you can adjust the number of prey you give your offspring according to the size of the prey. The other thing that might change is the abundance of natural enemies. So if natural enemies are common, then strategies like progressive provisioning might be optimal. But if you're in an environment, perhaps you're following one of these two nest first strategies, and natural enemies become rare, then perhaps you might get a reversal to prey first, which is the simplest strategy, perhaps least costly in terms of neural investment. That might do best. Yeah, actually, I might just try and include this. So just as an example, then, if we look specifically at these reverse transitions from nest first multiple prey, the yellow dots, to nest first single prey here and here, why might evolution have proceeded in that direction? So why, might you specialize on just giving your offspring one large single prey, even if perhaps they're single prey? And an interesting paper came out this year by Milena and Rosenheim that illustrates the sort of reason that backwards transition might happen. So they looked at these really weird creatures called stylops in the order Strepsiptera. Here's a couple of them here. They're tiny little things. If you've ever seen them, it's quite unusual to see them. They're only about 1 millimeter or a few millimeters long. And they have these, they produce tiny planidium larvae, really tiny larvae, which sit on flowers. And when an aculeate visits the flower for nectar or pollen, they can attach themselves to the adult and get taken back to the nest. And then they become endoparasites on the wasp or bee's offspring, internal parasites of the wasp or bee larva. Then when the aculeate larva becomes an adult, this is the tip of the abdomen of an Ammophila female. What happens is the stylops larvae inside pupate, and they protrude from the abdominal segments of the aculeate. And you've probably seen this if you've got lots of specimens. So I think this one is a male stylops pupa, and this is a female. The males hatch out as these strange adults and mate with the female, who stays in the abdomen of the host. But a key point is that an aculeate female that's parasitized like this is going to be sterile, so she can't reproduce. So in other words, from the point of view of the mother of this individual, this is a kind of total failure. This offspring might as well be dead. It's not going to be able to reproduce. And what Milena and Rosenheim did was they looked at the effect of the number of prey that you give your offspring on the proportion of your offspring that get parasitized. So each time a female wasp takes a caterpillar into her nest, she's risking introducing one of these stylops larvae that's going to effectively kill her offspring. And Milena and Rosenheim looked at about 9,000 specimens of 16 different Ammophila species in museum collections. And they recorded, because the nice thing is you can see these stylops pupae sticking out of the abdomen. So they recorded what proportion of the adults in the collection were actually parasitized by stylops. So on the x-axis here, then we've got the average number of prey that each Ammophila species, these were all Ammophila species, each Ammophila species gives its offspring. And on the y-axis, we've got the percentage of adults in the collection of each species that were parasitized. So each one of these dots here is a different Ammophila species. And you can see there's a quite tight relationship between the number of prey and the percentage parasitized. So the more prey you provide your offspring, the more chances for these stylops to get into the nest, the higher the percentage parasitism of your offspring. So 10% parasitized if you give your offspring about 10 prey, almost no parasitism if you provide only one large prey item. So this is the kind of enemy, and miltogramines are probably another one, that if it became common in the environment, that could favor a transition from multiple prey to single prey. Okay, finally, so I think the other thing this kind of thing emphasizes is the importance of natural history studies and museum collections. So, for example, this phylogeny is only part of the tree we've got for the species. And the reason I haven't included the other species in the tree is that we don't actually know what the nesting strategies are. So that's something where we need a little bit of natural history to find out what each species is doing. On the other extreme, there are species like these two European species, Ammophila campestris, Ammophila hungarica, where we know what the nesting strategies are, but we just haven't got a specimen in alcohol that we can get DNA from. So if you're ever in Europe and you see any of these two species, it would be great if you could put a male or a female specimen in ethanol. I'd be very pleased. Okay, so just to summarize that, there's lots of new phylogenies appearing all the time for the Hymenoptera, and they're interesting in their own right. They show the evolutionary relationships or hypotheses about the evolutionary relationships between different taxa. Obviously, they're only hypotheses, so as more data appears, they're probably going to change. But as well as that, there are lots of other interesting things we can use these phylogenies for. I've just talked about one of them, looking at what transitions in parental care strategies have occurred during evolution. Another thing we've looked at with the Ammophila phylogeny is whether ecology correlates with sexual dimorphism in specids. And there are lots of other things one can do as well. But important to say, though, this sort of thing really does rely on natural history knowledge to be able to map different traits onto the phylogeny and museum collections so that we can, for example, get DNA samples. To finish them, to acknowledge the other people I've worked with in these studies. So Martin Kennedy in New Zealand, I worked with on the phylogeny itself. And then for the parental care transitions work, it was Becky Bolton and Alejandro Gonzalez Voya. Okay, well, thanks very much.